Go. Okay, so these are the 4 steps on how DNA replication works in commixture form. So step 1 is the parent molecule has 2 complementary strands of DNA, and each base is paired by hydrogen bonding with its specific partner. So A with T and G with C. So just like it says, A with T here, G with C, A with T, A with T, G with C. So step 2 is the first step in replication is a separation of 2 DNA strands. So since they were together here with their specific pairs or partners, they separated here in step 2. So now with step 3, each parental strand now serves as a template or a temple that determines the order of nucleotides along a new complementary strand. So now they're like slowly starting to separate like one by one. And then now with step 4, the nucleotides are connected again to form the sugar phosphate backbones of the new strands. And each daughter DNA molecule consists of 1 parental strand and 1 new strand. So now you can see they came together and they're matched with their specific partner again. And that is how DNA replication works.
Okay, so these are the 5 steps of DNA replication. The first is helicase, the second is primase, and the third is DNA polymerase number 1. The fourth is DNA polymerase number 2, and the fifth is ligase. Okay, so a helicase is when it breaks the hydrogen bonds and like unzips the DNA. So right here, this little donut thing is like unzipping the DNA. And the SSBP is the single strand binding proteins, which temporarily keep the 2 DNA strands from coming back together. Number 2 is primase, which lays down in the RNA primer. So RNA is coming in here. Number 3 is DNA polymerase, which adds DNA to the 3-strand end of RNA. So it goes from being strictly RNA to the DNA replacing it on the 3-strand end. So it's now half RNA, half DNA. And then this together is called the Okazaki fragment. DNA polymerase number 2 replaces RNA with DNA, so it goes from being RNA and DNA to this DNA. And then it goes from, and then step number 5 is ligase, which attaches and breaks in the DNA backbone, which is a dehydration synthesis. So these 2 are separated, and then in ligase they come together.
Okay, so before, as Jordan explained, these are the 5 steps to DNA replication. I'm just going to go over some definitions to make it easier to understand. So helicases are enzymes that bind and may even remodel nucleic acids or nucleic acid protein complexes. There are DNA and RNA helicases. DNA helicases are essential during DNA replication because they separate double-stranded DNA into single strands, allowing each strand to be copied. Okay, so as seen in step 3, DNA polymerase, known as DNAP, is a type of enzyme that is responsible for forming new copies of DNA in the form of nucleic acid molecules. Nucleic acids are polymers, which are large molecules made up of smaller repeating units that are chemically connected to one another. So this is when the parental template DNA strands replicate and the daughter strands are synthesized in 2 different ways, which would be leading and lagging. So leading strand is formed continuously in a 5' to 3' direction, and the lagging strand is formed in sections, which are called the Okazaki fragments, in a 5' to 3' direction. So Okazaki fragments are short, newly synthesized DNA fragments that are formed on the lagging template strand during DNA replication. They are complementary to the lagging template strand, together forming short, double-stranded DNA sections. So now I'm just going to explain what the origin of replication is, the parental strand and the daughter strand, and then the 3 end and the 5 end. So daughter strands are the complement of the parent strands. So as you can see here, the parent forms a template for the daughter strand, and because of the way they are formed, in the DNA, because the DNA polymerase attaches complementary nitrogenous bases, example like thymine or T or adenine for A. And the daughter strand will also be what is called anti-parallel to the parent strand. So the 5 side of the parent strand will match with the 3 side of the daughter strand. So if the parent strand runs 5-3, the daughter strand will run counter 3-5 when matched up with the parent strand. Okay, so the origin replication, also called the replication origin, is a particular sequence in a genome at which replication is initiated. This can either involve replication of DNA in living organisms such as prokaryotes or eukaryotes, or that of DNA or RNA in viruses such as double-stranded RNA viruses. Yeah. Okay, so the 5 and the 5 end and the 3 end mean the 5 prime and the 3 prime, which indicate the carbon numbers in the DNA's sugar backbone. The 5 prime carbon has a phosphate group attached to it, and the 3 prime carbon has a hydroxyl group. So this is the dehydration synthesis of a carbohydrate, and just 2 things to know about dehydration synthesis is that it forms a bond while losing a water molecule, and that when 2 glucose molecules bond through dehydration synthesis, water or H2O is taken out. So as you can see in the diagram right here, it starts off with an H2O, or it starts off an OH on this side and an HO on this side, and then it comes down and it loses the water molecule that it had before. So step 1, water molecules removed from adjacent OH groups, and then 2, the remaining oxygen joins the 2 monomers with an oxygen bridge. So relevant to DNA, nucleotides are organic molecules that serve as the monomers or subunits of nucleic acids like DNA and RNA. The building blocks of nucleic acids, nucleotides, are composed of a nitrogenous base, a 5-carbon sugar, ribose or deoxyribose, and at least 1 phosphate group. Okay, so this is a picture of DNA, which is also a double helix. And a double helix refers to the structure formed by double-stranded molecules of nucleic acids, for example DNA here. The double helical structure of a nucleic acid complex arises as what? Okay, so this is a picture of a covalent bond. The empty circle represents an electron from hydrogen, and the colored in circle represents an electron from carbon. And a covalent bond involves a sharing of a pair of valence electrons by 2 atoms, in contrast to the transfer of electrons in ionic bonds. Such bonds lead to stable molecules if they share electrons in such a way to create a noble gas configuration for each atom. So here, I'm just going to explain what a replication fork is. And a replication fork is a site on a DNA molecule at which unwinding of the helices and synthesis of daughter molecules are both occurring. So you can see like the daughter strand here, the parental strand here, and then the replication fork. The replication fork shows the simultaneous synthesis of both strands, the daughter strand and the parental strand. Since synthesis occurs in the 5 to 3 prime direction, so it would be like 5 prime, 3 prime. And then, yes, that is it. Topoisomerase is a class of enzymes that alter the supercoiling of double-stranded DNA. The topoisomerases act by cutting one or both strands of DNA. Topoisomerase type 1 cuts one strand, whereas topoisomerase type 2 cuts both strands of DNA to relax the coil and extend the DNA molecule. This picture is an example of topoisomerase type 2.
Thanks.